Imagine a world free from genetic diseases. CRISPR, a revolutionary gene editing tool, is making this future possible. It allows scientists to precisely alter DNA sequences. Join us as we explore CRISPR's impact on medicine. Our genes dictate our health, and CRISPR offers unprecedented control over them. Acting like a molecular scalpel, it corrects genetic defects. From cystic fibrosis to cancer, CRISPR offers new hope. Let's delve into the mechanisms behind this technology. CRISPR's origins lie in bacteria, where it acts as an immune system. Bacteria capture viral DNA snippets, creating a genetic memory. CRISPR guides the Cas9 enzyme to cut viral DNA. This defense mechanism is now a powerful gene editing tool. Scientists have harnessed CRISPR-Cas9, a precise gene editing toolkit. The Cas9 enzyme cuts DNA, guided by a specific RNA molecule. This guide RNA directs Cas9 to the exact DNA sequence. The cell's natural repair mechanisms then introduce genetic changes. CRISPR enables precise gene editing by exploiting the cell's repair pathways. NHEJ can introduce random mutations. HDR uses a DNA template for accurate edits. This precision offers control over the genetic code. In 1987, scientists discovered mysterious repeating DNA sequences in bacteria. These sequences, later known as CRISPR, puzzled researchers for years. They were part of a bacterial immune system against viruses. This discovery laid the foundation for CRISPR's revolutionary role in genetics. In the early 2000s, researchers cracked the code of CRISPR's function. They found that bacteria use stored viral DNA to recognize and defend against viruses. This adaptive immune system was harnessed for precise gene editing. CRISPR's potential for targeted DNA modification was realized. In 2012, Jennifer Doudna and Emmanuel Charpentier's work on CRISPR-Cas9 revolutionized gene editing. Their discovery allowed precise DNA cuts, transforming life sciences. CRISPR's applications in medicine, agriculture, and biotechnology are vast. Clinical trials promise to transform healthcare. CRISPR's potential lies in correcting genetic defects. Diseases like cystic fibrosis and sickle cell anemia could be cured. CRISPR offers hope for treating complex genetic diseases. This technology could revolutionize treatments and prevention. CRISPR offers a new weapon against cancer, targeting cells with precision. CAR T-cell therapy uses CRISPR to engineer immune cells to attack cancer. CRISPR enhances the effectiveness of existing cancer treatments and enables personalized therapies. CRISPR offers hope for treating inherited diseases by correcting genetic defects. Gene editing in embryos could prevent diseases from developing. CRISPR is also being explored for treating diseases in children and adults. This technology promises to transform lives affected by inherited diseases. CRISPR's potential extends beyond gene editing to diagnostics, drug development, and agriculture. It offers rapid, accurate tools for detecting diseases and genetic mutations. CRISPR is transforming agriculture by developing resilient and nutritious crops. Its applications are vast, pushing the boundaries of science and technology. The ability to edit the very essence of life, our genes, is a profound power that comes with immense responsibility. CRISPR, with its unprecedented precision in manipulating the building blocks of life, has ignited ethical debates worldwide. As we stand on the cusp of a gene-editing revolution, it is crucial to carefully consider the ethical implications of this powerful technology. One of the most pressing ethical concerns surrounding CRISPR is the potential for unintended consequences. While CRISPR offers remarkable precision, it is not perfect. Off-target edits, where CRISPR inadvertently modifies genes other than the intended target, can occur. These unintended edits could have unforeseen and potentially harmful consequences. Another ethical concern is the potential for genetic enhancement. While CRISPR holds enormous promise for treating diseases, it could also be used to enhance desirable traits. Such genetic enhancements could exacerbate existing social inequalities. Moreover, the use of CRISPR in gene editing raises questions about consent, particularly in germline editing. Who has the right to make decisions about the genetic makeup of future generations? These are complex ethical questions with no easy answers. Perhaps the most contentious ethical debate surrounding CRISPR centers on germline editing. Unlike somatic cell editing, which targets specific tissues or organs in an individual, germline editing involves modifying the DNA of sperm, eggs, or embryos. Changes made through germline editing are heritable, meaning they are passed down to future generations. Proponents argue it holds the potential to eradicate devastating inherited diseases. They believe it is unethical to withhold a technology that could prevent debilitating diseases. However, opponents argue that germline editing crosses a fundamental ethical line. They raise concerns about unintended consequences for future generations and society. Germline editing also raises concerns about genetic diversity. Altering the human gene pool could have unforeseen consequences for our species' resilience. It is crucial to proceed cautiously, engaging in open and transparent public dialogue. Section 3. Ensuring equitable access. CRISPR for all? As CRISPR-based therapies become a reality, ensuring equitable access to these potentially life-saving treatments is paramount. The high cost of developing and delivering gene therapies could exacerbate existing health disparities, making these treatments accessible only to the wealthy. The ethical principle of justice demands that everyone, regardless of their socio-economic status, has equal access to healthcare, including gene therapies. Addressing the high costs requires exploring innovative funding models and ensuring fair pricing strategies. Equitable access to CRISPR therapies goes beyond affordability. It also requires addressing disparities in healthcare infrastructure, education, and information. Efforts are needed to ensure underserved communities have access to genetic testing and treatment centers. The promise of CRISPR hinges on our ability to ensure its benefits are shared equitably. This requires a collective effort from scientists, policymakers, healthcare providers, and the public. Victoria Gray, a young woman from Mississippi, had endured a lifetime of pain and suffering from sickle cell disease. This inherited blood disorder, caused by a single gene mutation, leads to the production of abnormal hemoglobin. Sickle cell disease is a condition where red blood cells become misshapen and can block blood flow, causing severe pain and complications. The mutation affects the hemoglobin, the protein in red blood cells that carries oxygen throughout the body. In 2019, Victoria volunteered for a groundbreaking clinical trial using CRISPR to treat sickle cell disease. This trial aimed to correct the genetic mutation responsible for the disease. The trial involved using CRISPR to modify a gene that affects fetal hemoglobin production. Fetal hemoglobin is a type of hemoglobin that is usually only produced during fetal development, but can be beneficial in treating sickle cell disease. The results were remarkable. Victoria's body began producing high levels of fetal hemoglobin, which helped to alleviate the symptoms of sickle cell disease. The pain crises that had plagued her for years vanished, and her overall health dramatically improved. She experienced a significant reduction in the frequency and severity of pain episodes. Victoria's case became a beacon of hope for millions worldwide living with sickle cell disease. Her story highlights the potential of CRISPR technology to transform lives and offer new treatments for genetic disorders. Timothy Ray Brown, the Berlin patient, made history as the first person cured of HIV. Diagnosed in 1995, he managed HIV with antiretroviral therapy. A bone marrow transplant from a donor with a rare mutation cured both conditions. Post-transplant, his HIV levels remained undetectable. Timothy's case offers hope for millions living with HIV. The CRISPR revolution is still in its early stages, but its impact on medicine is already profound. As research progresses, we can expect to see even more innovative and transformative applications of CRISPR. One exciting area is CRISPR-based therapies for complex diseases like heart disease and Alzheimer's. CRISPR's ability to target multiple genes simultaneously makes it promising for these diseases. Another avenue is using CRISPR in regenerative medicine to repair damaged tissues. For example, regenerating heart tissue after a heart attack or repairing damaged nerves. CRISPR-based diagnostic tools are advancing rapidly. These tools offer faster, more accurate, and affordable disease diagnosis. The journey ahead holds potential to reshape the future of medicine. The journey through the world of CRISPR reveals a future brimming with possibilities. This revolutionary technology can precisely edit the code of life, reshaping medicine. From curing inherited diseases to personalized therapies, CRISPR is transforming healthcare. Victoria Gray's triumph over sickle cell disease and Timothy Ray Brown's cure from HIV highlight its potential. Yet, we must acknowledge the ethical considerations that accompany this power. Responsible innovation and open dialogue are essential. The CRISPR revolution is a testament to human ingenuity and our pursuit of knowledge. Let us embrace CRISPR responsibly, ensuring a future of hope and well-being for all. As we stand on the brink of a new era in medicine, the potential of CRISPR is vast. Imagine a world where genetic diseases are a thing of the past, where personalized medicine is the norm, and where our understanding of biology reaches new heights. The possibilities are endless, but with great power comes great responsibility. We must navigate the ethical landscape with care, ensuring that the benefits of CRISPR are accessible to all and that we avoid unintended consequences. The future of medicine is here, and it is up to us to shape it wisely. By fostering collaboration, encouraging innovation, and maintaining a commitment to ethical practices, we can harness the full potential of CRISPR for the betterment of humanity. Join us on this journey into the future of medicine. Subscribe, like, and share this video. Thanks for watching, and we will see you in our next video.